Hello guys. Welcome to my channel. Today we are going to discuss about reproduction in lower and higher animals. So, very first, what is reproduction? Reproduction is a production of young ones like their parents. It is a biological process. There are two methods of reproduction. Asexual method of reproduction and sexual method of reproduction. First, asexual reproduction. It is a production of genetically identical progeny from a single organism that is from single parent. It does not involve fusion of two gametes. Fragmentation, budding and spore formation are some ways of asexual reproduction. And when plants reproduce asexually through their vegetative parts, it is known as vegetative reproduction, for example cutting and grafting. Now, sexual reproduction. It involves fusion of two gametes. In plants, a female gamete is ovule and male gamete is pollen grain, whereas in animals a female gamete is ovule and male gamete is sperm. It is characterized by fusion of male and female gamete, formation of zygote and embryogenesis. This mode of reproduction are grouped into three stages: pre-fertilization, fertilization and post-fertilization. In plants, reproduction is carried out in flower. And the male reproductive whorl of flower is called as androecium and female reproductive whorl is called as gynoecium. Now we discuss about structure of anther. It is a part of androecium. It is dithyx that is having two lobes and tertasporangiate that is having four pollen sacs. A mature anther is consist of four layers, namely, epidermis, endothecium, middle layer and tapetum. So, epidermis is an outermost protective layer of anther. Next to epidermis there is endothecium, which is a subepidermal layer. Then 1 to 2 layered thin middle layer. And then tapetum, which is innermost nutritive layer. There is presence of sporogenous tissue which is act as a microspore mother cell which divides mitotically to form haploid pollen grains. Then microsporogenesis. Microsporogenesis is the formation of microspores, that is pollen grains. It is carried out in anther. The sporogenous tissue of anther divides mitotically and form microspore mother cell. This microspore mother cell further divides meiotically and forms haploid microspores, that is pollen grains. The developed microspore or pollen grain is non-motile, unicellular and uninsulated cell, which is surrounded by two layered walls known as sporoderm. The exene is an outermost thick layer and intine is an inner layer of pollen grain. Then there is presence of germ pore. It is a thin area present on exene which helps in formation of pollen tube. Now the development of male gametophyte. During this, pollen grain starts to divide mitotically. After this first meiotic division there is formation of two cells, namely vegetative cell and generative cell. Vegetative cell is bigger in size whereas generative cell is small and thin-walled cell. This generative cell further divides mitotically and produce two non-motile male gametes. This two male nuclei then passes through pollen tube and reach up to embryo sac, that is female gametophyte, for fertilization. This remaining part we will discuss in our next video. Thanks for watching.